Okay. Hello and welcome to Water Conditioning Probiotics webinar. My name is Lucia Barreiro and I am the editor of Hatchery Feed Management. This is the fourth in the series Trends in Aquaculture Hatcheries that we are hosting, focused on different topics of importance for hatcheries. One of the main bottlenecks in hatcheries is the high unpredictable mortality and disease management. Probiotics and water conditioning are alternative strategies to manage diseases in the early stages that have attracted extensive attention in recent years. This webinar will focus on how to improve larval performance through probiotics and microbial management. Thanks to the support of our sponsors, INVE Aquaculture and Lallemand Animal Nutrition, this webinar is open and free. Each speaker will give a 15-minute presentation and then we will get to a final panel session to answer questions from the audience. To submit a question, please type it in the Q&A box. Especially thanks to our assistant editor and webinar manager, Marisa Yanaga, who is making sure all our webinars work. And finally, it's my pleasure to introduce Ruben Props, who will be moderating this webinar. Ruben is co-founder of KYTOS, a Ghent University spin-off company that develops microbial fingerprinting technologies to create actionable microbiome insights. He's specialized in the field of single-cell analysis. Currently, he is working on establishing the definition of a healthy microbiome in shrimp aquaculture, as well as understanding the importance of microbiome variation across aquaculture ecosystems. Ruben, I hand it over to you.
Thank you, Lucia. So hello to everyone listening in from around the globe. It's truly my pleasure to moderate today's webinar on a topic that I feel is instrumental to the aquaculture industry, as well as being close to my own interests. Out of all the challenges facing the aquaculture industry today, microbial diseases have remained the #1 area of concern. As I'm sure both the speakers and the audience have experienced, there remain vast uncertainties on the relationship between the microbiome and how it affects disease and health of the animals we cultivate. I'm therefore delighted today to present you with 4 titans of the industry with a combined experience of over 100 years, if my math is correct, all of whom are actively seeking to develop solutions for microbiological challenges in our industry. I'm sure we all have much to learn, and I'm looking forward to moderate this session. Before we start, some small technicalities. Each speaker will present a 15 to 20-minute presentations. Questions can be given through the chat box, and we will be part of the Q&A at the end of the presentations. I would also like to encourage the audience to actively send those questions, as we will do our best to incorporate them at the end of the Q&A session. That being said, without further ado, I would like to introduce our first speaker of today, Tania De Wolf. Tania graduated as a bioengineer from Ghent University in Belgium, where after she joined the R&D department of INVE Aquaculture 25 years ago, with the task of developing products for live food and marine fish diets. In 2001, she became R&D Manager of INVE's Aquaculture Research Center, located in Tuscany, Italy, where she leads the research group that works on fish projects. She is also responsible for the development of live feed cultures and enrichment products at INVE. Today, she'll be talking about the importance of microbial management during the larval rearing of sea bream aquaculture. Tania, the floor is yours.
Thank you, Ruben, for the introduction. And of course, I want to thank all the organizers of this webinar in order to give me the opportunity to talk to you this evening about the importance of the microbial management during larval rearing of sea bream. Just a short introduction. I think everyone knows more or less that the annual sea bream production in the Mediterranean area is the largest of the marine species cultured here and is around 700 million of fry per year. The production is yearly increasing with around 6%, and also the survival rates are increasing. So this is of course indicating that the culture is getting more intensive and we should be careful about the possible disease outbreaks that can happen. Just a short intro about the challenges in disease control. I think everyone knows very well this graph, which is showing the interactions that there are between 3 main factors. So the environment, the pathogen, the host, so in our case, the fish, and those interactions can decide whether or not a disease is happening. So there are a lot of factors in each of these 3 compounds, and I'm not going to sum up everything, but I will just focus on the blue ones that are also the topics that are covered a little bit in this presentation. So I will talk on live food disinfection, so mainly for the reduction of Vibrio, and I will talk about the use of probiotics, immunostimulant, and other nutraceuticals that are used in enrichments. So let's have a look to the importance of live food in this story. I think we are all concerned and we know very well the importance of the nutritional factors that we have in our live food enrichments and the importance on the performance of the larvae. But it's even sometimes more important what we bring to the larvae in terms of microbiota through the live food, because the larvae have a closed mouth, and the first encounter with the microbiota, with the bacteria that are present in the environment, is when the mouth is opening and when they start really the exogenous feeding. And this importance can be even more important than the nutritional factors of the live prey. So what we can do is, of course, that we need to take care of the quality of the live food that we are offering. And how can we do this? There are many parameters for this and many ways to take care of this. And I will focus only on 3 of them because we have very limited time. So first of all, I will talk about disinfection of the live food, then the use of probiotics in order to get the microbiota under control, and the use of last generation of enrichment products that are including also several nutraceuticals. So I will share now some results of the live food studies that we have done. First example is the disinfection of the rotifers. So in this case, we were working with Sanacare Sure, which is a water conditioner, and it's based on herbal ingredients, all natural ingredients, and this can be used after the enrichment of the rotifers or after the culture. And the main goal of this is to reduce the Vibrio counts. As you can see in the pictures with the platings and also in the graphs, Vibrio counts are decreased considerably when we use this disinfectant on the rotifers. A second example is what happens if you use a good bacteriostatic during the hatching and enrichment of Artemia. So I expressed here the bacterial load at the end of the Artemia enrichment. So platings done on marine agar and on TCBS to have the counts on Vibrio, and comparing the control treatment with the bacteriostatic treatment. And we can see here that there is mainly an effect on the reduction in Vibrio load. A side effect that is often observed is also the increased viability of the Artemia naupli when we are decreasing the bacterial load. So it's also important, or it's giving us the opportunity, if we reduce the bacterial load in our live food, we should be careful, of course, that we don't give too much space to fast-growing opportunistic bacteria. And by the use of probiotics, this can be done. So the probiotics can be used or through the treatment of the live food, or it can be used directly in the larval rearing water. The probiotics which we worked with is a Sanalife MIC-F, and this is a mixture consisting of 3 different bacillus strains. I'm just going to show some results of the use of these probiotics. So we applied these probiotics in 2 different ways. In the graph later on, we will have a legend, and ER plus S is referring to the use of the disinfectant and the probiotics in the rotifer harvesting tank, so in the live food. PB in W refers to the direct application of the probiotics in the larval rearing tank. And we also have, of course, negative control where no probiotics were used. Then we did a larval rearing trial with sea bream for 30 days, and we analyzed the sea bream larvae at 15 days post-hatch. This in order to have mainly an idea of what is happening during the rotifer feeding phase. And we were determining the bacillus that were present in the gut microbiome of the sea bream larvae. This was done to 16S Illumina sequencing, and the number of reads of bacillus are shown here in the graph. So what we see is that we have quite good effect of the use of probiotics. We find back the bacillus that we have in our probiotic mix inside the sea bream larvae. And the effect is bigger when we use the probiotics through the live food compared to the addition of the probiotics in the larval rearing water. And also our negative control was fine because we didn't have any bacillus reads in this negative control treatment. We also studied the use of nutraceuticals included in the enrichment for both rotifers and Artemia. So this is included in our last or most recent enrichment product, which is Easy Dry Salco. I'm not going to show you all the details because of time limits, but I'm referring to the presentation that I have done in EAS in 2016, where we could show that by using this enrichment product containing these different nutraceuticals, we can stimulate the immune system of sea bream larvae in very early stages. So we did these controls through molecular gene expression, and we saw that there was upregulation for different genes that are involved in anti-inflammatory processes or antiviral responses and so on. Then what we did is combining all these different pieces of the puzzles into a larval rearing protocol for the sea bream. So trial description, sea bream larval rearing trial that was done in 500 liter tanks. We stocked around 100 larvae just hatched per liter, sorry. And we used the green water technique. Rotifers were fed between day 4 and day 28 post-hatch. Artemia and dry feed were fed from day 19 to 55, so a classical larval rearing protocol for the sea bream. We controlled the stress resistance of this larva in 2 different time points, one at 30 days post-hatch, so at the end of the rotifer phase, and one at 56 days post-hatch, which is near to the end of the weaning. And then, of course, we did the end evaluation at day 60, where we also determined survival rate, growth rate, and we also determined the malformations that were present in this sea bream larvae. The treatments that we have done was, we had, of course, the control treatment referred to as COM in the next slides. And both control and treatments were done with 4 replicates. Here we used a standard enrichment product, and we didn't care about microbial management of the rotifers, neither of the Artemia. And then we had our microbial control treatments referred to as micro, where we enriched the rotifers with Easy Dry Salco. We applied our disinfectant, and we applied our probiotics. For the Artemia, here we used bacteriostatic during hatching and enrichment, and we used same enrichment, Easy Dry Salco. Now we're gonna have a look to the results. So if we look to the survival rates at day 56 post-hatch, we see that there are no main differences in average survival, which was quite good, 27%, but we see that there is a lower variation in survival in the micro treatment compared to the COM treatment. If we look to the growth that we obtained, we see that the weight, individual wet weight at 56 days post-hatch was bigger in the micro treatment compared to the COM treatment. And as a consequence, with a similar survival rate, we achieved a higher biomass, tank biomass in the micro treatment compared to the COM treatment. Another interesting results that we saw was in the stress resistance. So we applied a salinity stress test. We exposed the larvae to an increased salinity at the end of the rotifer phase, so 33 days post-hatch mainly. And we recorded the mortality during a period of 60 minutes. Every 3 minutes we read the mortality and we were summing the mortality in order to get the mortality rate. And as you can see in the graph, the green line is representing the micro treatment. We can see that we have a lower mortality in the micro treatment compared to the COM treatment. We repeated this at the end of weaning, and even at that time, we could observe the same trend. So also at the end of weaning, we still had a lower mortality during the stress test for the micro treatment compared to the COM treatment. Sorry. Regarding the deformity level, so we did studies through histology, and we were focusing, I represented all the severe deformities, the orange bars, and the main deformity that occurred in the sea bream larvae were operculum deformities. So we see that in the COM treatment, we have higher number of deformities compared to the micro treatments. So operculum deformities were reduced from 9.3% to 2.3%. Total deformities from around 16% down to 4%. And another very interesting result was shown at the level of the farm gate. So we were following these fish that were treated only during the first 60 days of life, and we followed them up to the growth and their performance in the cages. And what we did to evaluate the stress level, the chronic stress level that was accumulated during their lifespan in fish of 350 grams. This was done by Professor Johanna Arts from Stresscoron. And we determined the cortisol level that was accumulated in the scales, so representative for the whole lifespan. And also this was a very interesting results because we could see that in the micro treatments, they are represented by the green dots, we had in those 20 fish very similar levels of scale cortisol, while this was much more variable in the COM treatment. So lower levels and also more consistent levels of cortisol. So as a conclusion, we can summarize the importance of the microbial management during larval rearing of sea bream. And we could observe that it's important and to promote a favorable gut microbiome in the larvae. In our case, this was proven by the presence of the bacillus. We have more resistant and more fast growing larvae in our population, and we have a better fry quality. And in general, we also have a more predictable hatchery output. And I conclude with this. Thanks.
Thank you very much for a wonderful presentation, Tania. I'm sure we'll come back with many questions at the end of the session, but we have to move on to our next speaker, which is David Kawahigashi. David has 40 years of technical and management experience in all phases of shrimp aquaculture. He is also the founder and director of Vannamei 101, which is an aquaculture consulting and training company based out of Hawaii. And his areas of expertise include the design, as well as the implementation of multi-phase intensive production systems, as well as genetic programs and hatchery management as a whole. Today, he'll be talking about the importance of ultrafiltration and the use of symbiotics in the hatchery. David, please, the floor is yours.
Okay, good morning, everyone. Can you see my screen okay? Yes.
Yes, perfect.
Okay, great. Okay, well, first of all, thank you for tuning in, everyone. And I wanted to thank the organizers and sponsors for the opportunity to be here. What I'd like to do is discuss two areas of hatchery management that are not very common, but I believe are a growing trend. And that would be the use of ultrafiltration and the use of symbiotics in the hatchery. Basically, it's a dual strategy where ultrafiltration would effectively eliminate any pathogens, be it bacteria, spores, viruses from the water entering the hatchery, followed by an outcompete or a competitive exclusion model where you would repopulate the ecosystem of the larvae tank with a healthy microbiome using symbiotics and biofloc. So I'm going to cover these two topics. I think the application or use of ultrafiltration systems coincided with the arrival of EHP, this microsporidian parasitic fungus that has really impacted the industries globally, really. And this is a slide from back in 2015 where in Vietnam, a farmer said they would prefer that the shrimp die than have slow growth due to EHP. And of course, they'd rather cut their losses and start again rather than just raise the shrimp, feed the shrimp that are not really growing. And then come to present day, 2020-21, it seems that EHP continues to be a nemesis to the hatcheries, where in many countries, the majority of the ponds today are infected with EHP. So it's a very difficult disease to eradicate because of its nature of reproduction through spores. And I think that is why ultrafiltration has come on the scene, because it's a technology that works with other industries such as desalinization of water. And by utilizing this technology, they can effectively eliminate the EHP microsporidian spores from the water coming into the hatchery. There is a technique to reduce or remove microsporidians from the hatchery once it's in the hatchery, and that would be to do acid and alkali rinse of the water system, which is not as efficient and can delay or postpone interrupt in production, as opposed to ultrafiltration, which is a continuous cleansing of the water. And I think this technology is borrowed from the reverse osmosis desalinization process for turning seawater into potable drinking water. And Singapore is a good example where the majority of their water is coming from ultrafiltration units, or I should say reverse osmosis desalinization units. And so the process is fairly straightforward. It all begins with microfiltration, and this is a level that most hatcheries are using, where they would mechanically filter the water from the sea into a series of filters to clean the water, removing most of the turbidity and sediments, followed by ultrafiltration, which effectively removes the microorganisms, bacteria from the water, nanofiltration, which goes down to 0.001 micron, and then to the reverse osmosis unit, which removes the minerals and turns the seawater to drinking water. So our interest is in this ultrafiltration level right here. Ultrafiltration benefits are several. There is a good cost benefit to using the system, because essentially you're eliminating all pathogens coming in with the water. It's not to say that they won't come in other means, like with algae, with the feeds and maturation, etc. But at least the water that's coming into your hatchery system is clean or free of pathogens. So that's a step up. And the hatcheries that do use the ultrafiltration units, at least the ones I know in India, they're not treating their water after the ultrafiltration with chlorine or ozone or permanganate because they feel it's not necessary, and they have a history of not having or not detecting these pathogens in the hatchery after the ultrafiltration system. So that brings a benefit to, say, to enhancing the reproductive performance of broodstock in maturation, because the residuals sometimes from these treatments can affect the spawning performance of the broodstock in maturation. So as the spores are approximately 1 micron in size, the ultrafiltration can effectively remove these spores from the water. And the steps are fairly straightforward, depending on your source of water. You can bring it in directly from the ocean through slow percolation sand filters, or if you have well points, pumping water from beneath the beach. That pre-filtered water can go into the next step, which would be the rapid sand filter bag filters, followed by activated carbon filter, the multi filter for removing metals and iron, and then into the ultrafiltration unit, followed by UV, I guess, as a backup treatment, and into the hatchery. Okay, this is just a slide on activated carbon, how it would further polish the water and remove some of the organic, dissolved organics from the water before entering the multimedia iron removal filter, which consists of several types of media to filter and remove suspended impurities and minerals out of the water. Followed by, of course, water entering the ultrafiltration permeable membrane, which filters down to 0.01 micron. And all of these units, when you do order these large systems, they come with an automatic backwashing system, because when the pressure in these permeable filters reaches a certain limit, the system would automatically backwash using acid and alkali solution to clean the filters so that they could function properly. So it's pretty sophisticated from what I've seen. But yet, only a minority of the hatcheries, especially in Asia or everywhere, are using these ultrafiltration systems. And as you can imagine, the main reason why is because of the cost of investment for these systems. We're talking for a 50 cubic meter per hour ultrafiltration UV disinfection system, $100,000 plus $60,000 for industrial UV disinfection, tax, and installation costs. You're talking about a $200,000 investment for one hatchery, but it's good for 200 to 300 million PLs per month. So, there could be a cost benefit. There would be, given the situation of the quality of your larvae that you're selling out of the hatchery. So there's more interest in these ultrafiltration units. If you go on Alibaba.com, IndiaMart, Google, you can see many companies that are selling smaller units. There's only a few selling these mega units for hatcheries, but at least you can find a lot of information on these types of filtration systems and effectively clean up your water to the next level. So now that your water has been sterilized and disinfected, no pathogens, the next step would be to repopulate your water using beneficial bacteria and a healthy microbiome. And this, typically the probiotics is the standard practice for trying to develop this healthy ecosystem and microbiome for the hatcheries, for hatchery water. But what I would like to do is discuss the use of symbiotics and biofloc as another means of getting that microbiome up and creating stability in your water quality as quickly as possible. And as hopefully most of you know, symbiotics is a synergy between a complex carbohydrate such as rice bran, soy, wheat, etc., fermented with probiotic bacteria, beneficial bacteria, creating a fermentation solution or that would help to stimulate the microbiome in your water larvae tanks. And as you know, this fermentation process, the metabolic pathways of fermentation is very complex. It's not only bacteria that you're multiplying, you're creating a variety of different metabolites, bacteriocins, amino acids, peptides, etc., that is reacting with the water, the bottom, the animals themselves, modulating their internal health. So it is beneficial to people, to all organisms, and to shrimp. So I am a proponent of fermentation. Rice bran, fermented rice bran has been used directly in larvae tanks and maturation systems with good results. It's not that common, but it continues to be used. Of course, the solution is filtered through a 100-micron mesh because it's really just a liquid that we're trying to add to the tanks to create that stability in the water. Fermented sweet potato along with fermented soy has been used in hatcheries with good results. And even tapioca, cassava, manioc has been used as a source of complex carbohydrates. And this is just an example of how the survival of host larvae being treated with fermented rice bran has increased. This is, I think, from Ecuador. But one area of interest that it's even new to me is that they're now using biofloc symbiotics to create what we call a mature water tank and a mature water reservoir. And typically in a larviculture cycle, in the later third of the cycle, when the PLs reach about PL4 to harvest, is when you're doing your most water exchange. And the hatcheries usually use clean, sterile water to do this water exchange. But now hatcheries are beginning to use biofloc symbiotic water to do the water exchange because you have less impact on the stability of the tank at that point. Instead of adding clean water, you're adding mature, stable biofloc symbiotic water. And I believe this will be a tendency going forward. This is a standard protocol for preparing mature biofloc symbiotic water. It's a combination of organic carbon coupled with the addition of inorganic nutrients such as ammonium chloride, sodium nitrate, buffers such as sodium bicarbonate and soda ash to keep the pH and alkalinity high. So these mature biofloc reservoirs can be maintained for months, even up to a year, as long as you keep the nutrients and the nitrifying bacteria healthy. So I think this is a trend going forward. And in case you're curious to know what is the recipe or formula for fermentation of prebiotics, this is the one that is quite common using rice bran, but you can use other types of prebiotics to create this symbiotic culture to add to your tanks, larvae or grow out. And you can, I guess, take a copy of this when you have their presentation. But that is to me, the two areas that I think will be the drivers to the hatchery industry, it's a combination of low technology, being symbiotic biofloc systems, coupled or combined with essential tools to help the hatchery operator better manage the conditions of the hatchery and the diseases, using technologies like RIS, IoT, artificial intelligence, ultrafiltration, and to quantify the microbiome, of your tank system. So I think going forward, it's these two areas that are going to be, I guess, further developed in the future. So that brings me to my last slide. Thank you very much for your attention, and I'll hand it back to Ruben.
Thank you very much, David. It's really nice to see that these membrane technologies are slowly making their way also into the aquaculture sector for water reuse and for customizing water quality. Really nice work. Thank you. So next speaker is Stephane Ralite. Stephane is the Aquaculture Product Manager of Lallemand. He has solid experience and knowledge of the aquaculture segment in different regions around the globe, among which he has raised shrimp himself in Thailand, and he has also held several positions within Evialis INVE Group in France, where he played a key role in the setup of its aqua division Ocealis. After being aquaculture market manager within Diana Group for its Aquatif division, he set up his own consulting company where he brought his aquaculture expertise to different organizations. And since 2014, he has been in charge of the global development of Lallemand Animal Nutrition aquaculture activities worldwide. And today he will share his experience on the effect of probiotics and bioremediation in modulating shrimp and water microbiota in shrimp production units. Stephane, please, the floor is yours.
Oh, you're muted.
It will work better like this.
Perfect.
So, hello everybody, and thank you very much, Ruben, for the introduction, and thank you also for the first speaker for the interesting presentation. The objective today is to speak to you about microbial management in shrimp hatchery and nursery. As time is short, I will concentrate on only one trial that we did on real production units in Vietnam following microbial population in water and in the shrimp, and following the impact of bioremediation and probiotic bacteria that we added in the system. And for this, I want to thank also François Cellier, Anna Odiles, and Nguyen for their contribution for this work and presentation. So before going to the trial, for those who don't know the company, a very short introduction of Lallemand. So we are specialized in yeast and bacteria production for more than 100 years now. We have 4,500 employees. We do only yeast and bacteria, and we have 45 plants, including 29 fermentation plants. So we ferment our own microorganism, which allow us to select the strain, the process, etc., to make it specific to the targeted application. And we are present in all the market, able to use yeast and bacteria, which allow us to work on the understanding and and the manipulation of a very diverse microbial ecosystem. So it goes from control ecosystem, like for the food application, for example, production for food, or the production of food, like the bread, the yogurt, the cheese, the alcohol also for human, or for biofuel for the car. But also the study and the manipulation of live gut microbiota for human, for livestock, for shrimp or fish, or also the work on natural environments for the microbiota of soil, for agriculture, of water, for aqua, or also the biofilm, for example, the surface development of microbial population on the surface, like in hospitals, for example, after disinfection, You want to control the microbiota that comes. So in all these microbial ecosystem, a major difficulty is to identify and follow the different microorganisms present. And it's even more difficult in natural and open systems like soil for agriculture or water for shrimp. But thanks to so new technology, I should say, thanks to democratization or availability of technology, which are actually not so new, we are now able to follow much deeper, complex microbial ecosystem. So to illustrate this, I will show you a practical example in shrimp hatchery and nursery production unit. But before this, I want to give you a short introduction about amplicon sequencing, which is a genomic technique that we used to analyze the microbial population. So what is it and why is it useful for in this experiment? So usually in shrimp farming, we use cultural or plate counting technique to analyze microbiota. But to do this, you have to target specifically one presumptive bacteria, genus or species, among the total community. And you can target only a few of them. Amplicon sequencing is a more global approach as it allows to observe the whole bacterial community. You are analyzing DNA from the whole bacteria population. And here, for amplicon sequencing, we target a specific genes which could be considered as the ID card of the bacteria and is commonly used, it's the most commonly used marker, so it says 16S RNAG. So this technique allows us to better respond to some of the frequently asked questions that we can have on the farm on microbial management, especially in shrimp farm. So, for example, does water microbiome impact all life stage similarly? Can we track bioremediation and probiotic bacteria in situ, in water or in the gut of the shrimp? Do bioremediation and probiotic bacteria reduce undesirable bacteria? And similarly, do bioremediation and probiotic bacteria increase beneficial bacteria? And are all bioremediation or probiotic products alike? So the experiment was done in Vietnam. We started in a hatchery, concrete tanks with nuclei, so 200 nuclei per liter. And then after 22 days, we transferred the PL10 to the nursery tank at 2 post-larvae per liter. And then we raised them for 25 days in the nursery. The protocol, so we had 2 different protocols for the hatchery and the nursery. The 2 treatments in hatchery, the Lallemand protocol, and the control treatment was antibiotic oxytetracycline used in water. The Lallemand protocol, we had LALC-PL pack. So LALC-PL pack is a mix of Bacillus and Bactocell, Pediococcus acidilactici, so specific strain of Pediococcus and Bacillus mixed together, applied in water. And at the same time, we used a probiotic, which is Bactocell, so in the field. For the hatchery, we had 3 treatments. The Lallemand treatment, the Lallemand protocol, so with LALC-PL Bacillus plus Pediococcus acidilactici in water. Lalpac Probio, which is a specific pack with Bactocell plus a specific yeast, Saccharomyces boulardii, so a proprietary yeast of Lallemand that we mix together in the feed. And then we had a treatment with a control treatment without any bacteria, and another treatment with competitor protocol with product containing also Bacillus and Pediococcus acidilactici, but of course different strains. So first question, did water microbiota impact all life stage similarly? So on this graph here, each dot represents the microbiota characteristic, let's say, of one sample. So if the dots are close, the sample, the microbiota composition is similar or close. If the dots are far from each other, the microbiota composition is very different. So you see that in the hatchery, if you look to the hatchery water, you can see that the water and the gut microbiota are very similar. This is actually quite logical, as in the very early stage, at zooid stage, for example, the mouth open and the first thing entering the gut is the tank water. So water microbial composition will have a strong impact on gut microbial composition at this stage. But if you want to have some impact on the gut microbial composition, the water microbiota should be controlled. In the nursery, when the gut microbiota and the shrimp immunity are more mature, there is a clear differentiation. Here you see that the gut microbial population is different from the water microbial population. And this differentiation is growing with time. If you look to the grow out phase, for example, it's even higher. So here the action on the gut, direct action on the gut microbiota should be done through the feed. It will be much more efficient. Other question, can we track the bioremediation bacteria or probiotic in situ? As it's different, and this is a question that we have frequently, because it's not always easy to answer globally with the actual technique used on the farm. So the answer is yes. We follow the Bacillus and Pediococcus population in the hatchery and the nursery. As a reminder, Bacillus and Pedio are the bacteria contained in the product that we added in the system. And we found that in the hatchery and in the nursery, we had more Bacillus and Pedio in Lallemand treatment compared to the control. Did bioremediation bacteria and probiotic reduce undesirable bacteria? The answer is yes. So here you have a graph representing LDS score, so linear discriminant analysis. This allows to compare a group of microbes between two treatments and show, for example, the major differences linked to each of the treatments. And here you see that we find a high proportion of what could be called undesirable bacteria, like, for example, Algoriphagus. So I take some example which has been shown to be linked with EMS, HPNB, Vibrio through horizontal transfer, or Tenacibaculum, which is a well-known aquaculture pathogen, especially in fish. And you see that in antibiotic, you had much more proportion of these two, for example, compared to Lallemand, which is quite paradoxal because you are using antibiotic to control the possible pathogen instead of increasing. You have, with the bioremediation probiotic usage, potential impact on undesirable bacteria. Same in nursery water, we reduced 25 times, we had 25 times less undesirable bacteria in the Lallemand treatment compared to the control. And if you look to the biggest difference here, the Vibrio, you see that the biggest difference between the two treatments is the Vibrio. You have much more treatment in the control group compared to the Lallemand protocol. So I'm going fast here. What about beneficial bacteria? What do we call beneficial bacteria? It can be beneficial for the environment, or it can be beneficial for the shrimp. For the environment, for example, it can be the control of the nitrite, the toxic compound like H2S, or the soil quality. For the shrimp gut, it can be improving digestibility, immunity of the shrimp, or for both, controlling the pathogen, for example. Did biorem bacteria and probiotic increase beneficial bacteria? Yes. In the hatchery, for example, if we increased here Thalassobius bacteria, which are being considered as a probiotic candidate, synthesizing vitamin B12, troponidetic acid, which are linked to Vibrio antagonism or can improve also feed digestibility. If we look to the nursery shrimp, we also increased beneficial bacteria, as an example, Amaricoccus, which can store short-chain fatty acid with anti-inflammatory effect. You can see also that it increased the bacillus levels. And in the water, nursery water also, to finish quickly, we increased also with the Lallemand protocol compared to the control beneficial bacteria like Rubivirax, which are involved in nitrogen carbon fixation, or Caistobacter, which degrade toxic aromatic compounds. So we see very quickly that we can at least manipulate gut and water microbiota with the addition of specific bioremediation and probiotic bacteria. But are all bioremediation and probiotic alike? The answer is no. I remind you that in the nursery competitor treatment, we had also Bacillus and Pediococcus acidolactici, but of course different strains. And you can see clearly that we were able to recover Pediococcus in Lallemand treatment, but we find almost nothing in the competitor, and it was the same for Bacillus. So this shows clearly the importance of the strain. Knowing the species is not enough to understand the efficacy. And you can see some examples issued from our screening platform, which we used to screen microorganisms. It's also a bank. Here, just looking at the shape of the colony, this is 2 Bacillus subtilis, but you see that the colony looks very different, and it's linked to the enzymatic activity and also biofilm formation property, for example. Another example is on the enzymatic activity. Here you have the results of one result of amylase activity screening comparison between different bacteria. And you see that these bacteria are all Bacillus subtilis. But you see very clearly that comparing the amylase activity of this one, which is very high, it's where you can have some Bacillus subtilis with almost no amylase activity. So the strain makes all the difference. And if we come back to the experiment, and especially in the nursery, we can see that there were a clear difference of the impact of between the impact of the competitor product and the Lallemand treatment on beneficial bacteria population. Like, for example, Rugiaria, Paracoccus, or Phyobacter that we classified in the beneficial bacteria. If we look to the potential pathogenic bacteria, we had also a clear difference with much higher proportion in the competitor treatment compared to the Lallemand treatment. So I go a little bit quickly. We have seen that it is possible to see concrete impact on shrimp microbiota and in water, but what about the performance? It's what's of course interesting the farmer. If we compare the weight, the survival, and the FCR, we have numerically better results in the Lallemand group compared to the competitor. But what I find even more interesting here is the consistency of the results. In the Lallemand group, the 4 replicates gave much more consistent results compared to the control, where we had some good but also some bad results. And in hatchery and nursery, technician knows very well that the most important is to maintain consistent and predictable results between tanks and cycle. So as a conclusion, I will come back to a very practical question. It is clear that maintaining a stable gut and water microbial population is crucial, but how can we do this? And especially to work on the shrimp gut microbiota. What has a direct impact on the gut microbiota? So it depends on the stages. In hatchery, all the different microbiota population in contact of the shrimp will be, of the larvae, will be important, especially at the very early stage. So in the shrimp, it's like for the fish, for example, live food, as explained in the first presentation, will be crucial. The microbiota composition of the live food will be crucial. So enriching the Artemia here will have a very positive impact on the gut microbiota control. But the water microbial population, especially at the very early stage when the mouth opens, the microbial population of water will be also very important to control. And the feed, if you put the phytoplankton microbial population, quality, of course, so avoid contamination. But if you put some probiotic in the feed, you will have some direct impact also and being able to secure the gut microbial population. If we look to the nursery stage, we have seen that there is a differentiation between the gut microbial population and the water microbial population. So working on the microbial water microbiota is still very important because you can have a small impact on the gut microbial population, but also, of course, because the microbial composition in water is crucial to maintain a good water quality and equilibrium. So working on the water microbial population is important to maintain a good environment. But if you want to act directly on the gut microbiota, you should act through the feed, through the artificial or natural feed. This will have a strong impact on the gut microbiota. And if we look to the grow-out phase, so it's even the differentiation between water microbiota and gut microbiota is even higher. So you have to work on the water microbial population to stabilize the water quality. But if you want to act on gut microbiota, then feed and soil actually will have more impact, direct impact on gut microbial population. So the quality of the soil and the use of probiotic in feed will have a strong impact. And of course, don't forget, yes, the importance of immunity in stabilizing gut microbiota. So as I finish my time, the last slide will be some example. If you want to securize the gut and microbial population, you can use some probiotic through feed to have some direct impact on the gut microbiota. At hatchery level also, you should enrich the artificial live food with probiotic because it's the best way to bring it inside the gut. And it's also a good security for the live food production. And then you should use some barrier remediation bacteria also to control the water quality and to have some positive impact in gut. In nursery, the probiotic would have some impact on the gut, and the bioremediation in nursery, and especially even more in grow out, will be targeting water and soil quality. Time is over. So I thank you very much for your attention, and I'm available for any question.
Thank you very much, Stephane. So this was a really interesting presentation, impressive results. What I really liked is that you gave an overview of the in-depth characterization of a microbiome and showing all of us how complex the reality really is of the microbiome. But despite that complexity, we are still able to modulate it, which I think stems us very full of hope that we can actually have an impact on this microbiome and control it. So really awesome results. Thank you very much.
Thank you.
Then we move on to the crescendo of this webinar. So the final speaker, which is Steven Newman. Steven started his career in the commercialization and the development of the first commercial fish vaccines, as well as rapid diagnostic tools. Since 1996, he runs his own company called AquaIntech. He has consulted extensively for shrimp and fish farmers in dozens of countries, focusing on issues affecting sustainability, primarily related to animal health. For the last 4 years, he was also lead auditor for the Aquaculture Certification Council, and today he will also talk about probiotics for the hatchery. Steve, the floor is all yours.
Okay, well, hopefully I'm here and everybody can hear me.
Yes, we can hear you.
Next thing is to get this up here. Okay. Let's see here. This was down here. Do this. And then do this. Okay, so everybody can see this now?
No. We can't see your screen.
Can't see my screen. Okay. All right. Well, not sure why. Let's try this again here. Let's see. Allow. Interesting, because it's showing me the video's on. Let's get rid of this stuff here. Stop video, mute my video.
Make sure you share your screen, the green button.
Yeah, well, for some reason or another, when I click on Zoom, what it's doing, here, let's see. Let's try this. There we go. Let's do this one right now. That
There
should
we
do
go.
it. There we go. I got it. Now all I got to do is make it so that you guys can see the only thing that's important here. Let's see. There we go, I hope.
Great, perfect.
Okay, sorry. Basically, what I'm going to talk about today, just briefly, is probiotics for the hatchery. I'm going to focus initially just on definitions and talk a little bit about our experience in the field. I'm not really going to talk about mechanisms, specific mechanisms. I try to keep things quite general. I'll be talking a little bit about where these might be applied in broodstock or hatchery and nursery. I'm not going to be talking about our farm experiences. The first thing I want to really do is just start out with defining some of the terms. I find that it must be extremely confusing for some of the laymen out there, who are trying to follow what's going on, and a lot of different terms are being bandied about, and some of them are being used interchangeably. And frankly, I can understand why some people would be confused by this. I think one of the most important things, though, initially is to define what an actual probiotic is. This is something that's typical of what we see when terms get used outside of the original intended definition. They tend to evolve, and what's happened with the term probiotic is that it no longer really matches the definition that WHO, FAO, United Nations, and a few of the early workers in the field had sat down and established. And there are really four aspects of this. One is that in order for it to be a probiotic, it has to be a living microorganism. This is usually bacteria that can be yeast, fungi, or even others. It has to be administered orally. That is, it has to enter the animal by ingestion. It's not something that can be taken up by the gills. It must colonize the gut, and it must have an impact on the microbiome. It also has to have a beneficial impact on the consuming animal's health. This is a bit vague. It's not really clear what we're talking about here. We're talking about disease resistance. We're talking about tolerance. I think a lot of times it's interpreted as meaning to increase disease resistance. So as I mentioned, what's happened is that the term has sort of become a little bit different. It's being used in a different way than it was originally intended to be used. So the widespread usage is very broad. And nowadays, if you go out in the fields and you look and see what people are selling as probiotics, you'll see that most of these products actually have little or nothing to do with animal health. Typically, they are products that act via bioremediation or bioaugmentation. But the way the word's being used is it literally encompasses any application where living bacteria or fungi are involved. Personally, my own experience, based on what I've read in the literature, I think that true probiotics are actually rare, at least based upon this definition. And for shrimp, I think that it's going to be challenging at best to do this in the field. It may not even be possible. The reasons for this are complex. I'd be glad to talk to anybody at length about this. I think one is the plasticity of the microbiome. And I think also too, a lot of non-scientists and scientists confuse correlation and causation. So oftentimes what we see in the lab doesn't translate to the field either. So you can see things that work great in the lab, but when you go out in the field, they don't work the same way. What we do know though is that the intestinal tract of the shrimp is what's in the environment. So I put this little picture here to show you that when we talk about probiotics, we're not actually talking about yogurt here. The bacillus and other species that are in use act through a variety of mechanisms. Some of them probably, and I'm not, again, you saw slides with lots of potential ways that this might be occurring. You know, I think most of what we're seeing in the field, at least in the hatchery, is related to nutrient competition and limitation rather than the production of antimicrobial peptides and bacteriocins, which typically work the best when the bacteria in biofilms. So what you have here is a slide. This is actually someone else's. It's just sort of become generic, I think, in some sense, and that shows how these materials might interact with pathogens in the animal and the environment to bring about a desired impact. There is some speculation that there might be some immune stimulation that occurs here. I don't think that's necessarily the case in the field. It's complicated. Shrimp consume large amounts of bacteria normally as part of their diets. So we have to be careful about leaping to conclusions. I think that really under most field conditions, the mode of action of the product is through the use of bioremediation and bioaugmentation. So bioremediation truly is just simply defined as the treatment of pollutants or waste, like an oil spill or contaminated groundwater, via microorganisms. Typically, we think of bacteria that break down these undesirable substances. You can see a photograph here of a pond here that's in dire need of soil bioremediation. These black areas typically are accumulated areas where the soils become anaerobic. You've got hydrogen sulfide issues, and which have an adverse impact on the animal, and both direct and indirect. So this is what most, if not all, the products are being marketed as quote unquote probiotics for aquaculture do. They actually act via bioremediation. The pollutants we're talking about are accumulated organic matter from feces, uneaten feed, decaying plant matter, and so on and so forth. Bioaugmentation, it sort of overlaps. It's actually a form of bioremediation. And it's the addition of bacteria and similar organisms to accelerate the degradation of the contaminants. So it doesn't necessarily mean that the bacteria or the organisms you're adding become stable components of the ecosystem you're adding them to. Microbiome is an assemblage, let's see if I can go backwards here. Microbiome is an assemblage of microorganisms that colonize specific ecological niches. The microbiome concept is actually a very elegant concept. It's still evolving with time. One of the things I think that we are learning is that there's a lot more for us to learn. It's extremely complex. It's not a simple matter of just tweaking a few things and having permanent changes, especially in environments that are open, such as the average shrimp hatchery or maturation facility or even a farm, which is actually open in the environment and has a great number of uncontrolled inputs and which make it very difficult to come up with the concept of a stable microbiome. We started working with these bacteria about 16 or 17 years ago. Frankly, I was very skeptical about whether or not there was any ability to utilize these things. And I had been paid as a consultant to go in and take a look at some systems of pond-site culture that were being sold at that period of time. And what I noted was that most of the people were growing up Vibrios and Saccharomyces and an occasional Bacillus. It wasn't as straightforward as a lot of people thought it was. When we went out in the field and we tested an approach using a product which was actually patented for use in catfish farming, we found that it worked, but it didn't really work as well as we thought it would. And after a little while, I started thinking, well, why don't we just make something that goes right to the bottom of the ponds and the bottom of the tank? That's where the action is. And so we tableted the product. And when we did, we found we had some good benefits here. Again, I'm talking largely about hatchery experience. This is what my clients are telling us. It's not, they're noticing excellent control of TCBS, yellow, green, and LB colonies. Basically, in English, this means that they're seeing lower levels of Vibrios in the systems. They're also finding that the animals tend to grow a bit faster than they do in the control tanks, and they feed more aggressively in these tanks. They see less molting problems, fewer dead animals. There's much less fouling. We have some clients who comment that the tanks are virtually clean and free of bacteria. They're not free of bacteria, obviously, but this is a visual observation on their part. They almost universally observe lower levels of waste on the tank bottoms. And in some cases, it appears we actually get biofilm development in the bottom of the hatchery tanks just by adding these bacillus. They also note very low levels of ammonia, nitrite, and so it does impact the water chemistry. We have some clients who've actually been able to close their tanks and not exchange water at all. They don't need any other bacterial amendments. We've also done some testing where we've looked at the impact on Vibrio loads. Having done my PhD thesis on a Vibrio and that affects salmonids, I probably know a little bit more about Vibrio than the average person does. And you know, there's a lot of dogma in the industry. I think it's unfortunate, but the focus on Vibrio is excessive. There are many people around who have a simplistic view of what's actually going on, and they'll tell you if on a TCBS plate, if it produces an acid and causes yellow colonies, that's no problem. If a Vibrio on a TCBS plate can't do that and the colonies are green, you know, then they're all bad. And it turns out this is an oversimplification, and in fact, this focus on Vibrios, I think, tends to allow people to ignore some of the other potential problems. It's not, they'll just look at one thing. I'll look at the impact on Vibrio loads, and then they think that means everything's going to be great, when that's not necessarily the case. This is from a hatchery. Basically, they used our tablets in the hatchery, and this is a single replicate. This is not multiple tanks. They they did a plate count on TCBS and noted yellow colony counts. You can see that over the course of the application of the tablets, they saw a reduced Vibrio load. And then this was an example where the tanks where they were exchanging water. They had to exchange water because what they noticed was that in the controls, you can see that the bacterial loads get fairly high. They'd flush to reduce them. And Whereas in this tank setup, they didn't actually need to do that. And you can see, I mean, even though they don't go to zero, the bacillus were able to outcompete the Vibrios one way or another. Again, I'm not going to speculate entirely on the mechanism. I think it's personally through bioremediation. We're just taking nutrients away from the Vibrios. This is an impact on in TCBS auger. We're just looking at green colonies. Vibrio parahemolyticus, which is one of the nastier shrimp pathogens. It's the etiologic agent of EMS or aphids. Also, there are strains that are quite nasty that kill human beings as well. But not all strains are pathogenic. However, they're all green for the most part on TCBS. But you can see here, again, similar thing. They noted reduced Vibrio loads within 24 hours of the first application. This was daily application of the product at roughly 1 to 2 ppm. We have some clients who use it slightly higher levels, 3 or 4 parts per million. Everybody pretty much notices the similar type things. Recently, I just got some photographs a few days ago, that not in time for this presentation, that shows the tanks are clean and healthy, happy animals at all life stages. You can see right here, though, with the controls, how the bacterial loads went up, and that they were forced to exchange the water, and in order to reduce it, that was the only viable way. Whereas with the tablets, they didn't need to exchange water at all, substituted for it. So my conclusions from this are pretty straightforward, and that is that the use of bacillus spores, and these are spores we're adding. The tablets do not contain nutrients. The nutrients in the environment are what the bacteria utilize. It has a reproducible beneficial effect. We see a load reduction of potentially pathogenic bacteria. In this case, we focused on Vibrios. Universal comments among users is that the water quality is visibly improved. The spores have to be applied on a regular basis. They don't survive for an extended period of time in these particular environments. I think there's a number of reasons for that, but that's outside the subject of this particular presentation. I don't think the bacteria do much of anything, but they may become temporary members of the microbiome. And like I said, we have some indications that there may be some stable biofilm formation in some of these systems, that contain bacillus, but we have yet to do any extended testing to determine what actually is in these biofilms. The spores germinate in the presence of water, and they do this at rates that are species, strain, and environment dependent. So different strains germinate at different times, time intervals. Some of them take longer, some of them are much shorter. Some different species as well. Some may germinate, hatch within about 4 hours. Some may take 10, 12, 14, 18, 24 hours. Water temperature has a lot to do with it. Most of the strains of bacillus that are being used in this industry for this particular purpose globally are not saltwater isolates, they are freshwater isolates. They originated in wastewater treatment. When they're ingested by shrimp, the spores, they don't have time to germinate in the gut. The shrimp gut is short. Transition times are extremely short in the gut. In a small animal, it's in a matter of minutes from the time that food enters the mouth until the time it leaves. In a larger animal, it might be an hour, but that's not enough time for these spores to turn into metabolically active bacteria. So post-germination, that's when they're active metabolically. That's when they do their thing and actively digest organic matter. And as I indicated, the primary mechanism is through bioaugmentation and bioremediation. I don't personally think you're going to see stable colonization of the shrimp in a typical shrimp hatchery with any bacteria that we add. unless we are able to control most, if not all, the variables. There's just too many variables at play. And though, as I said, in a highly controlled production environment, I think it might be possible to do that. And that's the end of what I have to say. Thank you very much for your time and attention.
Excellent. Thank you very much, Stephane. So I think you touched upon many of the points that I wrote down for the discussion section. So it's a great timing to have you finish as the last speaker of today. So I think, Lucia, we can now move towards the discussion section and the Q&A.
Yeah, sure, go on.
Okay, perfect. So what I did throughout the presentations, I wrote down some overarching topics that we can touch upon in the discussion. And the first one that I wrote down is that is the importance of the taxonomic identity of the probiotics that people use, as well as the target location that we want these probiotics to be. For example, Stephane, you mentioned that do we want them to reach the gut, even though the transition time is very short? Do we want these probiotics in the water, in the particles, or in the sediment, or even in the biofilm? So I'd like to start the discussion with this, perhaps with Tania, to share your vision on what the target location is of the probiotics and how you select the taxonomic identity of the probiotics that you use.
Okay, so I think at least for larval rearing of marine fish, we have different targets that we want. We want, first of all, to just the occupation of space, so competitiveness in numbers, let's say. We also use them in order to aid the digestion, so by the production of exoenzymes. So this is also one criteria for selection. And maybe to a lesser extent in the larval rearing, but also the fact that they degrade organic matter can help, of course, in maintaining the water quality. So if you select strains, at least for us, this is very important to have a combination of strains that are combining all these different characteristics as good as possible.
And touching also on that, is that which trends do you see in that respect? Because I was reading up about probiotics. You also now have postbiotics that they say that are extracts of bacteria. So you don't need live bacteria anymore. Do you see any trends moving away from using live bacteria to other components to fulfill those functionalities?
Well, it would be maybe easier because it's very long and difficult to register and to get everything approved for the use of different probiotics. But I think it's a little bit of black box with still a lot of activities and are known, others are still unknown. So maybe we isolate some key characteristics that we can reproduce also by some extracts or whatever, but others are still a little bit a mystery. So I don't know if we can cover all the aspects that are covered now by the probiotics just by using some extracts.
Right. And Stephane, I saw on your website that you've also been working with bacterial extracts. I don't know if you want to comment on this as well. Stephane, you're muted, I think.
There we go. My early work with aquaculture was fish vaccine development, actually farm salmon, and my first endeavor in the shrimp industry was to see whether or not you could develop a vaccine for shrimp, this was long. This was, gosh, 30 some odd years ago, and so. At that time, there were like 4 papers on the subject, and it suggested that you could actually stimulate them non-specifically. When we found out that was the case, but and what we used was a bacterial extract. I mean, it was basically dead whole cell material, highly concentrated, so it could be used at a relatively high dilution, and it worked. It worked well. We stopped selling it after I learned more and more about the industry and realized that there were other things that were more important. And it was too difficult to get distributors to be honest about these things, because they would try to sell them as magic bullets and so, to solve problems that they could not solve. And you know, the farmers after a while would just start saying, well, it's not working. And it would be, well, that's because you're trying to fix something that you can't fix with these. But
Right.
yeah, so the extracts worked. We did. You know, so we're looking at possibly reintroducing the product as a parabiotic.
Okay, interesting. And this brings me actually to the big question: why does everybody choose to work with Bacillus as a probiotic? So I think maybe Stephane, and then David, you can also chime in, because you choose a different path with symbiotics, on what your opinion is on the use of Bacillus and how we can move beyond or complement this as a product.
Yes, so it's a good question. You have a lot of Bacillus product on the market, because this is the majority of the bacteria produced today, especially as Stephane explained, for water treatment, for example. There is a lot of Bacillus produced for this, and then that are sold also for aquaculture. Bacillus has also the advantage of being spore forming. So for bioremediation, it's for the stability, if it's useful, because the stability of the product is quite long. And Bacillus is a natural microorganism present in the shrimp pond, which is involved in organic matter degradation and ammonia, let's say the nitrogen cycle. So globally, this is some reason why it's available, and so the company are using it. But it's very true that, of course, in the nature, you have much more bacteria that are involved in. So if we speak about bioremediation, so the water, the environment quality, stability, let's say, there is much more bacteria than Bacillus. So looking for those bacteria and trying to develop them is important. In Asia, for example, you can find also some product with other strain than Bacillus. The problem is the stability of these bacteria. A lot of those products are, let's say, containing not so much livable or alive bacteria. But for sure, there is a lot more bacteria that are important in bioremediation, and same for probiotic. The problem is to have an effective probiotic, as pointed out by Stephane. You have to demonstrate that it's effective, that it's present, it is actually in the gut. You have to show it, and then that you have some impact. So Bacillus can have some impact, but we, of course, as it has been explained, the replication time of the bacteria is also important. So non-spore forming bacteria have an advantage, like in gut, in the action on the local gut microbiota, and the selection of the bacteria, specific selection of the bacteria for its real action. Not only looking at the FCR and growth on the farm, but understanding the mode of action, how does it work and why, is of course important.
For me, there's so many very good probiotics and commercial probiotics in the market. And I take a more subjective approach in that with symbiotics, we're ramping up the whole bacteria soup, so to speak, and meaning that if a product or probiotic is specific to a certain type of bacillus, it's not to say that after fermentation or even in the culture system, that bacteria is going to be the one that's dominating, or that's providing the the benefits that the probiotic brings. And so what I see symbiotics as providing is a more diverse blend of different multitudes of bacteria. And we're talking counts, after 24 hours of fermentation, in excess of 10 to the 9th. So it's quite high, and they're all reacting with each other. And so I take it as sort of the probiotic initiates the process, but after that, depending on the ecosystem of the pond or water quality or salinity, etc., certain types of bacteria may dominate over the primary bacteria that comes with the product. So that's sort of my point of view on that issue.
Yeah, excellent point, because this brings me to a question that several viewers also asked, is on dosing of probiotics. I saw both from Tania and Stephane that you showed results on how abundant the probiotics were in your trials, but several users or viewers have asked, what's the target dose of probiotics? Do you know, is there a target that you want to reach? X% of my community should be probiotics. I saw numbers on the presentations going from 0.5 to 5% even in relative abundance. Is this sufficient? So can both of you maybe comment on what your targets are on how much probiotic you want to introduce and colonize in the community?
Yeah. For us, we are calculating if we apply the probionts in the larval rearing water, for example, we are targeting to have around 10 to the 8 colony forming bacteria per milliliter of water. So this should be enough to theoretically have the whole population under control. So we are targeting this level.
Okay.
Yes, today it's more also in CFU per milliliter or gram, or that the target. We don't today think in percentage of the population. Maybe not yet, but for sure we are not still there. The global study in shrimp aquaculture is the global microbial population is still at the starting point, even if you begin to have a lot of publication. But so we are targeting also a specific CFU concentration in the feed, or to reach specific concentration in the gut and or in water, if this is the water that you are targeting, or enrichment media, etc. And this will depend, of course, on the bacteria. But yes, this is today, it's demonstrated efficient concentration in real situation.
Right. And is there something as dosing too much probiotics? If you dose too much, can you have adverse effects on the animals as well? Is this something that is known, or is there an upper limit that you would also say?
So it would depends on the bacteria used for our probiotics. So actually we have only one probiotic, and thank you, Stephane, also for the definition. Probiotic is active in the gut, live microorganism active in the gut, and we have only one, which is Bactocell. It's actually the only one which is registered as probiotic for aquaculture in Europe. So, sorry, can you repeat your question?
If there's a concentration or a dose that's too high for the animal.
Okay, too high. So, and we can there is no overdosing. Today we didn't see, or in normal utilization, of course, we didn't see some maximum dosing of Bactocell as a probiotic. For bioremediation, it's something else, because this is quite well known from the shrimp hatchery technicians. If you use, for example, some bacillus, some of them can have an impact on the algae bloom, and maybe David also can comment on this, or Stephane, they have a large practical experience, but you can have some change the equilibrium in closed high density system with the use of probiotics. So you have to be careful. Bacillus are heterotroph and aerobic. You can have an impact also on oxygen consumption. So for the water conditioning, you have to be careful, depending, of course, on the bacteria that you use.
There's actually, if you don't mind my interjecting here for a second, there's actually a peripheral issue that relates to this, and that is how these products are being marketed. You know, you have the science, i.e., you know, what are the best levels to use in a given environment? What's going to give you the best effect and the best impact?
Of course, of course.
But on the other hand, you also have an industry that's full of snake oil salesmen, you know, who are selling products, making claims that don't make any sense. I just saw a package in Ecuador where somebody was claiming they had 100 billion CFUs per gram, you know, in a product that they were selling for $20 a kilo. And so this confuses the issue, because what happens is a lot of people don't understand that, at least from the standpoint of the organisms we work with, it's about the food that's present. It's not about what you start with. As I explained to them, look, if I have the right nutrients, I can start with a single cell and end up with massive amounts of bacteria, you know, so more is not necessarily better. And I think, as Stephanie pointed out as well, you know, you can get blooms in some of these environments that are potentially problematic. I warn people about this at times, you know, especially if they're adding sugars and other nutrients to the system, that they can, in theory, overdose it. And you pointed out, and these aerobic bacteria will reduce the oxygen levels in these systems and cause problems that way.
Okay, I'm going to jump topics now because there's a very interesting topic on water quality, which I want to start with David on. So I wrote down two questions based on, or two takeaway messages from your presentation, is that we have two choices in aquaculture. Either we manage the water that we have to create the water quality, or we created it ourselves using membrane technologies. Is there, and which route do you prefer, or do you see that there's just a complementarity to them?
I think it comes down to maybe the severity of the disease situation in the area, because if you're in a country like Fiji and there's no shrimp pathogens, you don't have to do much treatment of the water or filtration even. But if you're in you know, Southeast Asia, where all the main shrimp producing countries are, you have options to, as I presented, put in ultrafiltration systems, you know, make big investments, which the companies with deep financial pockets can do. Or you can go with the conventional treatment systems with disinfection. But as I mentioned, it only works for most diseases, but not the EHP. So you're going to have to deal with the EHP issue. So, I mean, it's, you know, your options and you know what the situation is in the hatcheries. And I think it just comes down to what you're able to, at what level that you're able to clean your water coming in. And it's not only water, it's also the management of the feeds you're using. The fresh feeds and maturation can bring in pathogens, etc. So it's I think it's a choice that the operator has to make, you know, as far as at what level of biosecurity they wish to implement in their hatchery. So like I mentioned, a minority of the hatcheries use ultrafiltration. The majority do not. But I think as diseases become a greater issue, then it's going to begin to trend toward more higher technologies.
And a question from a viewer from Patrick Sorgelos is, hi David, why UV treatment following the ultrafiltration? That should have removed the bacteria as well.
Yeah, I wish I had asked this question earlier, because I also wondered why you need to have the UV sterilization following the ultrafiltration if the ultrafiltration is doing the job. My guess is that maybe it's a backup. It's like to ensure that if anything does get through the ultrafiltration system, maybe they forgot to backflush the membranes. If anything does get through, I guess the UV would catch it. That's the only rational explanation I can make, is that it's more like a backup to the ultrafiltration system.
Right. So then a question for Stephane from Tom Scrope. So when you add bacillus-based products, do you find this throws off nitrification, but outcompeting slower growing nitrifiers, or are the production cycles too short for this to have an effect?
You can have some impact of bacillus on nitrification, yes. So it depends on the system. But we have demonstrated that by using bacillus, you first act on the ammonia level, and by acting on the ammonia level, you will have an impact on the nitrification population, bacteria population. So you can also have an impact on the nitrification process. But bacillus will not do nitrification directly, at least the one that we are using. But you can have some indirect impact, let's say, on nitrification. But on hatchery closed system with a biofilter, the handling of the biofilter is of course it's much more important. So if you have a stable system, you can use bacillus in your system without any impaired impact on the nitrification process.
And maybe tying into that question from another question from Diana Chan. Whether these probiotics could also be used for other species such as mud crab and so on. So are these probiotics a one-stop shop across species, or do you have to have different probiotics for different species in aquaculture? And maybe Tania, you can also share your opinion on that.
Of course, you have to demonstrate that the probiotic that you propose is efficient. This is clear. The bacteria, we do all kind of screening, from the in vitro screening to the real on-farm application, going through research facilities. And you have to demonstrate that your probiotic is efficient in the bacteria, in the specific species targeted, of course. So this is what we do with the Bactocell, the probiotic that we sell. There is many publications on many species. But if we have a question on a new species, of course, it's important to test it in this targeted species. So the new species available are getting, there's not so much, but of course it is important, because the real in vivo application can always be specific.
Tania, maybe you want to share your view on this as well?
Yeah, I can agree with this approach, because also we are using bacillus strains that have a very wide tolerance both to temperature and for salinity. So salinity, they can withstand from 0 to 60 ppt. Temperature optimum is around 30 degrees, but also work at wider ranges. But we studied only the efficacy, so really looking into the host, if we can find back our probiotics that we are using only for bass and bream, tilapia, and the shrimp. So I think to be really sure, you should study also in the other species, of course.
Okay, thank you. Stephen, a question for you. I liked at the end of your presentation that you talked about the dogmas existing in the field, that it's really difficult to get away from these dogmas, giving a clear example on Vibrio plate counts that are used that are not really given sufficient nuance to perform microbiome management. And I was wondering on how do you see the industry breaking these dogmas and moving away from these dogmas and embracing other technologies and really advancing in that aspect as well. So I'd love to hear your thoughts, and everybody please chime in as well.
Well, I think that this is complicated. It's the analogy I sort of use when I think about this is when I first got involved in salmon farming, it was mom and pop. There weren't a lot of large companies doing this. It was small guys. It was guys, you know, that had a single net pen or half a dozen net pens. The industry in Norway was just tiny. It matured very rapidly. Within the decade or so that I was working with farmed salmonids, it went from being this mom and pop to being largely a corporate driven business. When I got involved in shrimp farming, the first thing I saw was chaos everywhere. And I figured that sooner or later market forces would act to force consolidation. That's happened, but it's very, very slow. Your question is an excellent one, and I've spent a lot of time thinking about this. And unfortunately, I can't be particularly optimistic about a time frame. I think one of the challenges in all of this is that most of the shrimp that are farmed in certain parts of the world are farmed by people who are poor and not educated. These poverty-driven paradigms are not consistent or conducive to consolidation. They're not consistent to the use of science in many cases. I can remember being on a farm in India not that long ago and talking to a couple of young guys who just got involved in the business and highly educated, we talked a little bit about what they were doing and what they knew and didn't know. And I commented about the cattle walking around their property and they're Hindus and it was, well, there's nothing we can do about this. And it's like, well, those cows are crapping in the ponds, and that can cause problems. Yeah, but there's nothing we can do about it. And I think in a lot of cases, that's what I see. I see people just going, what can I do? I can't really do anything. They don't have control. So I don't think in my lifetime I'm going to see much of what you're discussing being brought into reality. I think that we are going to see more and more people focusing on the science aspect of this, but with poverty-driven paradigms and literally huge numbers of companies that are selling anything and everything they can to these farmers and taking advantage of them in some cases, and too often in many cases, these things are going to have to change fundamentally. And even though I'm not a great fan of government oversight, in some areas, I don't see any other way for this to change. There are efforts being made now in some places, but it's 3 steps forward and 4 steps backwards in a lot of cases. It may look progressive, but in the end, when you go out and you take a serious look at what's going, it doesn't have an impact on the end users.
Yep. David, perhaps since you have 4 decades of experience in the aquaculture industry, I would love to hear your thoughts as well.
Yeah, thanks for revealing my age, Ruben. Hey, can you repeat the question again, please?
Well, the question was related to that there are a lot of dogmas in the industry with respect to what you can learn on the microbiome using plate counts and other types of management technologies that are very difficult to break or move past from. And so the question to Stephane was, what way do you see the industry respond and embrace new technologies and insights to break these dogmas?
Yeah, that's that's an area that hasn't really moved forward in decades, because when you do quantification of your bacteria, be it Vibrio or others, you're talking a 24-hour waiting time, for the colonies to grow on the media. And so there's no quick and easy way to do a water quality check and determine what your diversity is, or how to categorize the Vibrios or pathogens or good bacteria that's in your water. There's no quick fix. And I think that's where, technologies, what I mentioned in my presentation about new technologies would have to come in and try to fill that gap, because right now, the only option we have is to do microbiology agar plating. And that's not real time. So that's an area I think that hopefully can be improved in the near future.
And maybe coming back to the symbiotics, David, in terms of production, standardization of symbiotics, I feel like this is an important area that needs a lot of research because you're starting from natural materials. Do you feel that this is still a fruitful research area that requires still a lot of research, or is this battle proven already and tested How do you see the evolution of this type of product?
Yeah, good question. Yes to both, meaning if you look up what research and publications have been done on symbiotics, I mean, you'll be lucky to find one or two. And as far as the application of symbiotics as making it into a standardized operating procedure, it's not easy because it's so diverse. You know how culture systems are different, and then there's so many different ways that people are applying symbiotics. I mean, there's symbiotics for the water, there's symbiotics for the animal, for the gut to ingest. And there's ancient symbiotics called bokashi, which has been done for decades, where the fermentation of grains left in a pond to help to biofertilize the pond bottom and water. I mean, as I said, it's an ancient technology, but for modern day aquaculture, it's fairly new because not many people have heard or are doing symbiotic water treatments and animal treatments, because you have symbiotics that are meant to be coated on feed, and then you apply that to the pond, and the feed acts as a vehicle to bring the beneficial microbes to the animal to modulate the gut. And then there's the fermented rice bran, which is more like a planktonic type of water stabilizer or treatment, where you would use the metabolites produced from the bacteria during the fermentation process to help to stabilize the environment, the pH, the algae, etc. So no, there's no standard protocol. I just give a basic one. And typically what happens is the users that adopt the technology end up customizing it on their own anyway to make it conform to their farm. So in short, I think this is going to be a long-term trend, symbiotics. It's growing in countries every year that goes by. I see more and more people applying it. And I think we need to bring the science to the art, because symbiotics is still an art form. It's not so much science yet. So I'll leave it at that.
Well, I see people asking for your protocols in the chat. So people are very interested in trying it out, that's for sure.
Yeah.
The last big question that I have, that's an open question to anyone, is that I don't remember who mentioned it in their presentations, but biofilms. Biofilms are everywhere in nature as well, but we don't see them discussed very often in an aquaculture context. So in terms of probiotics and microbiome management, I would love to hear your thoughts on the importance or the non-importance of biofilms in aquaculture operations. So anyone, please feel free to give your idea or thoughts or experiences in this aspect.
Maybe I can start, because this is a field on which we are working not only for aqua, but as I briefly explained, this is for in hospital, for example, or in livestock or so, many different applications. Bacteria and certain bacteria are likes and prefer a mode of development is under biofilm form. And so if you want to control, and some pathogenic, for example, bacteria, if you want to control those pathogenic bacteria, you have to be able to control the formation of this biofilm. So the cleaning, the disinfection is very important, but then you have to avoid the development of new undesirable biofilm, bacteria biofilm. And for this, you can use, this is what we are working on and developing and already applying, some positive biofilm former that will form a biofilm which would avoid the formation of the, for example, Vibrio biofilm or Escherichia coli in other application, or etc. So the biofilm is a typical form of development of some bacteria, and controlling this biofilm is important. If we look to the gut, it's a little bit different, of course, but the way the development of the bacteria and for example, is it developing in the lumen, is it developing in contact with the enterocytes, etc., will make also a difference. in the efficacy and the impact of the probiotic. So this is a subject which is yes, very, very important. And of course, if we speak about hatchery and aquaculture and all the types, etc., this is biofilm is something which is commonly looked at by the technician, or should be.
You know, I think that biofilms have actually been underappreciated to a great degree in general. Quorum sensing is important. You know, microbes behave differently when there's just a few of them than there are large numbers of them. You know, there are going to be biofilms on animals and in animals and on the surfaces. Those biofilms are going to be impacted by inputs, and those inputs are going to be nutrients, they're going to be other microbes, they're going to be phages, there's going to be a variety of things that have an impact on the stability or the ability to generate stable biofilms in any of these particular systems. And it may very well end up being ultimately that developing technologies that allow us to make a strong, for lack of a better term, biofilm, you know, that can't readily be assailed by some of these pathogens, may turn out to be extremely important in controlling animal health and disease susceptibility. Eliminating pathogens is one approach, obviously, but you know, in shrimp farming, frankly, I consider that, unless you're in a highly controlled environment, to be impractical. You know, so
yeah, and this relates to a comment from a researcher also in the chat, where they said that in their PhD project, they saw that Ironomonas hydrophila also acted as a gut booster and a probiotic, contrary to most literature review that documents it as a pathogen. So even organisms that are generally considered pathogens, they also see sometimes positive effects on fish health.
Lots of Vibrios is that way too. There are Vibrios that aren't that are labeled as being pathogenic, but it's only until they, you know, form a biofilm that they begin to excrete or produce the toxins that become of concern.
Well, their question then is, and it relates to what Stephane was showing as well, so how do you really determine real hatchery pathogens when we only look at the identity of them?
Exactly. So this is one thing that I didn't develop here in the presentation. So it's we often in this presentation, at least, we often look, and until now we concentrate on specific pathogen and look if it's present or not, trying to find a specific pathogen for the causative agent of the pathology or the problem, and trying to concentrate on the absence or presence of this pathogen. But today we know it's not enough in its simplistic, of course, approach. There's many examples of this, but and you know it also well, Ruben and Tania, and as Stephane say, the Vibrio, just concentrating on Vibrio is not enough. One example, for example, is white feces. We don't know exactly the causative agent of white feces. We know that there is some agent impacting white feces development, and there is some consensus on the importance of some bacterial community that could develop the white feces, and of course the impact on EHP favoring these developments. But now it's clear that it's more important, if you want to have a stable production, to look at the equilibrium, to avoid strong microbial changes and disruption that would change this equilibrium and give opportunity for opportunistic bacteria to develop, for example. So this is why also the symbiotic approach with global stable microbial population is also interesting and has some logic. The problem, of course, is to control this microbial population, and ideally to find some parameters, maybe some ratio of families, Vibrio families, that could be marker of good equilibrium or entering a danger zone, for example, that could lead to some pathology development. But defining as I did simplistically, desirable and undesirable bacteria, of course, is not easy. There is some publication, some work. It is actually one of the topics today to try to find some ways to classify a good and safe microbial population or dangerous microbial population, to classify the parameters that can lead to problematic bacteria development. But this is actually research, actual work, and future work, which is very, very interesting and very important.
Yeah, definitely looking forward to seeing that work in the next presentation, Stephane. I have a final question, and then I've gone through all of the questions from the audience and also some of the questions that you posed and the ones I wrote down here. And it's for Tania. It ties into what we just discussed, is that in your presentation, you saw that microbial treatments are mainly related to controlling the pathogens, which in general expected are to improve survival. But what you observed is that there was no impact on survival, but there was an effect on the growth, stress-resistant deformity, so all of these nuances. So how do you explain then this effect of these microbial treatments? Do you know the causative mode of action, or can you talk a bit more about this?
Yes, so I think in this trial we didn't have any effect on the survival, because also it's difficult in the larvae to have real pathogens that are going to influence your survival rate. So if you have a good system where you have a stable, anyway, quite stable environment, you will not see any specific results on the survival rate. But we did see important effects on the growth, for example, and I think this is maybe not due to just one factor, but a combination of different factors. Also, for example, on the stress resistance, we know this can be caused by one of the compounds we have in our disinfectant product for the rotifers, and also, of course, by our nutraceuticals that are present in the enrichments there. Okay, the most common ones are astaxanthin, vitamin C, and so on, but also the plant extracts that we use there, and could be also an effect of the probiotics. So there are different causes. And also on the growth, better growth could be used, could be produced by, for example, the production of enzymes that where also the probiotics are contributing. So a better digestion, a better assimilation of the nutrients in the gut of the larvae, and so on. So it's a multifactorial effect, let's say, that we are able to observe.
Yeah, this again confirms that everything microbiome related is very complex, and we need to do a lot more research still to figure it out. So with that, I have gone through all of my questions, and we've discussed all of many of the points from the viewers. Lucia, I think I hand it over back to you to close out the session.
Yeah, I think it was such a great webinar. Thank you very much to all the panel for the great presentations and also for the great discussion. And thanks, Ruben, for the great job moderating. And thanks everyone for joining in. If you want to stay up to date on the latest news on aquaculture hatcheries, I encourage you to subscribe to our publications on our website, hatcheryfm.com. All our webinars are available on our website and on YouTube. See you next time. Goodbye.
Bye-bye.